Well, as I announced last week, basically, as of last Friday, we covered all the core material for the curriculum of this class. The last 2 weeks, we're just basically covering random topics that really I cannot do justice to any of these topics in a single lecture, so it's just going to be a brief introduction. But it's areas that in the past students have wanted to know stuff about so that at least they have the basic idea with respect to a certain type of statistical method. So if there's anything in particular that you would like to learn more about that's related to statistics, send me an email. If I don't have a lecture planned on the topic already, I'm happy to try to create one. I mean, I got a request at the beginning of the course to cover ROC curves, which I haven't covered in the past. I'm going to try to add that, but it will be the last homework, and I'll try to post the final as soon as I get a chance to write it as well, just to give everyone as much time to work on it as possible. It's going to be due basically at the end of finals week, and that is a fairly hard deadline just because a couple of people in the class that would like to graduate, and I need to have grades posted so that can happen. So anyway, without further ado, today we're covering principal components analysis. Like I said, this isn't something that normally gets covered in an intro course, and this is going to be a very quick overview by necessity, but it does get used enough in modern bench science work that I think it's worth knowing at least the basics about it, so that if you ever see it, you know what it is. So, what's principal components analysis? Well, this is kind of another method for dealing with the issue of multicollinearity that we've discussed fairly extensively, that say you have several x's highly correlated with one another. If you add more x's to a model, you get very little information because most of the information is already contained in the x's that you already have in the model. If you're doing regression, that can actually make things worse because when you add in more x's, that adds variance to your model, so it may actually decrease predictive accuracy rather than increase it because you get more variance but very little additional information about the outcome since it's so correlated with what's already there. And if you're doing like modern high throughput genetics, this problem's almost unavoidable. So, what can you do if you have a data set that's highly collinear? One option is to do variable selection. You try to find some subset that captures most of the information in the x's. Well, it can be really tricky to find the best subset of the x's that captures the most information, and the set of variables that you do find is very prone to overfitting, etc., etc. Another solution that we discussed is penalized regression, that you penalize regression coefficients that are too large, and that works pretty well in the context of regression, but it only works if our objective is to predict a certain y variable. What if we just have a bunch of x's, we want to try to reduce these x's down to a smaller set of variables that captures as much information as possible? We can't do that with penalized regression. Penalized regression, we have to try to predict something. Oh, one approach that's useful in this type of situation is what's known as principal components analysis, which is basically a way to try to reduce the dimension of your data set. Let's say you had a data set that looks like this. Normally, to describe the location of any given point in the scatter plot, you would have to give the x-coordinate and the y-coordinate, i.e. two different numbers. But you could notice looking at this data set that if you rotate the axes here, that almost all of the variance in this data set is on this particular axis. There's a little bit of variance in this other axis, but it's much smaller. So let's say, if rather than giving an x-coordinate and a y-coordinate, you only gave the coordinate of a point along this axis, like rather than giving an x-coordinate and a y-coordinate for that point up there, you just gave the distance along this red horizontal axis here. Now you'd be a little bit off, but you wouldn't be that far off. The coordinate of that point, you'd be a little bit further off, but for none of the points would you be horribly off. And by doing so, you've reduced the coordinates of each point from 2 numbers down to 1 number. Now, okay, if you go from 2 to 1, it's like, what's the point? It's not a significant improvement, but if you had like 100 variables, for instance, and you managed to reduce these 100 variables down to 1 variable or 2 or 3 variables that capture the vast majority of the variance in the data set, like we see in this figure, then you've made your data set much easier to handle. And that's the basic idea behind principal components analysis, to take a high-dimensional data set and reduce it down to a smaller number of variables that capture as much information about the larger set of variables as possible. So, in formal mathematical language, say you have a set of variables x1, x2 up to xm, and the idea behind principal components analysis is you try to approximate your x's as a linear combination of a bunch of principal component scores that are called S, such that you lose as little information as possible when you approximate the x's by these S variables. And these S's are known as principal component scores, and the A's are known as the loadings. Basically, you take the loadings, you take the scores multiplied by the loadings, and you get your approximation of X. And these S variables are chosen to have the following properties. The S1, S2 up to Sm are all independent of one another. More formally, they're orthogonal to one another. If you've taken linear algebra, statistically that means for all intents and purposes they're independent. And they're also chosen such that the first principal component has the largest variance, which is greater than the variance of the second component, and so on down to the mth principal component has the smallest variance. So, I mean, I know that's kind of crazy math here. What does this actually mean? Well, the first principal component is the one-dimensional approximation of the x's that captures as much information as possible about the x's. In the little graph that I showed a few slides back, when you rotated your axes such that the vast majority of the variance is captured on that one axis, that's the first principal component. It's basically, the intuition is that rather than two-dimensional space, it's n-dimensional space. You rotate your axes such that along the first axis, you get as much variance as possible about the x's. And the second principal component is the one-dimensional approximation of x that captures as much information about the x's while being orthogonal to the first principal component or independent of the first component. So basically, you put your second axis in a direction that captures as much variance as possible, but you make sure that you're independent of the first component. Then the third component is the direction that captures as much variance as possible that's independent of the first two components, and so on down the line. So how do you calculate principal components in R? Let's remember the Longley data set, where it's various measures or various variables that are believed to be associated with unemployment, and they're ridiculously strongly correlated with one another. So the command in R to calculate principal components is called prcomp. Notice in this case I just say twiddle dot date equals Longley, scale equals true. This twiddle dot just says use every single variable in the Longley data set to form principal components. If I had just said twiddle GNP deflator plus GNP plus unemployed, it would have calculated the principal components to only those three variables. If I just say a dot, it calculates the principal components to all the variables. And note that there's no predictor variable here. Principal components analysis doesn't try to predict a y variable. It looks only at the x's. So if you try to give a y variable to the left of the twiddle, it'll blow up on you. It only wants x variables. You'll also notice this command, scale equals true. That tells R to, before it calculates the principal component, it scales all the variables to have mean 0 and standard deviation 1. That's because if you don't scale it like that, the principal components tend to be dominated by the variables with the greatest variance, which usually isn't what you want. For some reason in R, its scale equals fault is the default command. They did it that way for historical reasons, but if you look at the help file for PRComp, it says you almost always want to do scale equals true. So I recommend whenever you use this command, you add the parameter scale equals true. If you print out the output, it gives the standard deviations and a rotation matrix. The rotation matrix is just the loadings that, in this case, if you want to get the principal component scores, you take GNP, the value of GNP deflator times this variable times this number plus GNP times this number plus unemployed times that number, and so on down the line. In practice, R will do as force. We don't need to worry about it too much. Standard deviations give us some idea of the amount of variance explained by each of the components, as I'll explain here in a second. If you've ever taken linear algebra, it turns out that basically the standard deviations are just the square roots of the eigenvalues, and the matrix of loadings are just the eigenvectors. If you have no idea what I just said, don't worry about it. It doesn't really matter for this class. And in general, if you say PRComp twiddle x1 plus x2 up to xn, that'll find the principal components of these variables. And if you say twiddle dot, that'll tell R to use all the variables in a specific data frame, but you have to pass the data parameter to tell which data frame that has the variables that you're interested in. And there's also no response variable y in this particular formula. Other way you can do is if you just have a matrix or data frame called x, you can just say PRComp x, and that will also calculate the principal components without having to mess with the formula. And as I noted earlier, when you use this command, my personal recommendation is that you always add the scale equals true parameter. Now, as I said before, the rotation matrix is the matrix of loadings, and the standard deviation measures how much variance is explained by each component, as I'll illustrate here in a second. If you want to calculate principal component scores, then you can use the predict command in R. Longley.pca is the output from the PRComp command. And here I say new date equals Longley 1, in other words, just the first row of the Longley date. I'm saying calculate the principal component scores for the first observation in this data set, which is the year 1947. Get the first score is minus 3.76, the second score is 0.682, and so on down the line. And yeah, in general, say predict of the output from a PRComp function. That'll give the principal component scores for each observation in the data that was used to calculate the model. And you can also give a new data command if you want to calculate it for specific x variables. These the points in new data may be part of the original data, or they may be entirely new points. And when you're using principal components, one of the obvious questions is how many principal components should you include in your model? And that's actually a tricky question. Answering it is as much art as science. But one of the common ways that people use to try to answer that question is to plot what's called a scree plot, as illustrated here. And the scree plot is just basically, you saw the standard deviations from the previous output. It's just the square of each of those standard deviations, and the height of each bar is just the square of the standard deviation of each component. So basically what this tells you, that's the amount of variance explained by the first component, that's the amount of variance explained by the second component, that's the amount of variance explained by the third component, and so on. And the folklore is, if you're trying to choose how many principal components to use, then you look for a quote-unquote elbow in this graph, which is basically the point at which you're getting very little additional variance once you go past that particular component. In this case, you can see that the first component is capturing the lion's share of the variance in the data. That's kind of to be expected when you have the variables are strongly correlated with one another as they are. In this data set, a second component captures a much smaller hunk of variance, but a still noticeable amount. The third component is capturing a very small amount, and 4 through 7 are capturing essentially 0. So for this particular data set, if you just used only the first component, you're not losing very much information at all. And it looks like if you go out to 3 components, you have basically 100% of the information that you would get from the full set of 7 variables. So I certainly wouldn't want to go past 3 in this case. You may want to go with 2 or even 1. I mean, like I say, there isn't necessarily a right or wrong answer here. So basically, went over this on the previous slide. The first component captures the vast majority of the variance. You get second and third that capture a smaller but noticeable amount. Fourth and beyond capture essentially nothing. You would want to choose between 1 and 3 components here, most likely. So how do you choose how many components? Well, as I said, the folklore is that you just look for an elbow in the scree plot where there's a huge amount of variance up to that particular component. Once you get past that component, you're not explaining much more additional variance. But, I mean, in the Longley example, that's reasonably clear-cut. In most other real-world examples, it won't be that obvious. And sometimes people use other criteria, like a common one is just taking all the components with variance greater than 1. Even though in practice that doesn't work that well, there's various other approaches that are more advanced that people have tried. If you're unsure about this, talk to an expert, because honestly, the problem of figuring out a number of components is still a research area, and I wouldn't say it's a solved problem at this point. And how do you plot this scree plot in R? Well, you just take the output of the PRComp function and pass it to the plot function. In this case, we call the output longley.pca, so we just say plot longley.pca, and we can give axis labels and so forth as usual. Easy enough, hopefully. In general, if you say plot of a PRComp object, that will give you a scree plot of this particular model. So, why would you ever want to use principal components? And that's a little bit of a tricky question because it's useful anytime that you would want to reduce the dimensionality of your data. So, I mean, up until now in this class, I've tried to lay out a relatively clear set of rules for when you would use a particular method. That if you have one continuous number, if you have continuous numbers in one of two conditions, case versus controls or whatever, and you want to compare the two, you can use a t-test for something like that. You could lay out a set of rules like that. When do you use principal components? Well, anytime it would be useful to reduce the number of variables in your data set. When would that be useful? It can be useful in a lot of different situations, and it's really hard to lay down a list of rules saying, if your data looks like this, use principal components. So I'll just describe a couple places where you might want to use principal components. And yeah, for practical purposes, if you ever use it in any of your lab work, then you already know what it's useful for, and if you don't, then you may not care about this lecture that much anyway, so yeah. But one possible use of principal components is in the context of regression. That, as we discussed in the past, if there's multicollinearity, that can cause problems when you fit your regression model. And we discussed various ways to do that. Stepwise methods, as horrendous as they are, still get used. There's also penalized methods, which can solve the problem, but another potential solution to this multicollinearity problem is to use principal components. As I said earlier, principal components are chosen to be independent of one another. So rather than fitting a regression model to the original data, we can fit a regression model to the principal components, and multicollinearity disappears as an issue because the principal components are designed to be independent of one another. So the idea is, rather than predicting y based on all the x's and letting multicollinearity blow up your model, you just predict y based on the first principal components and hope you can get decent predictions that way. So remember the Boston housing data, an example I used when we were discussing penalized regression. Say we want to determine which factors affect the price of housing in various neighborhoods in Boston. They collected data on 13 possible predictors of housing prices from 506 different neighborhoods, and the objective was to predict the median value of homes in each neighborhood based on each of these 13 predictors. And a couple weeks ago we showed how you can use lasso or ridge regression for this type of problem. Now I'll illustrate how you can tackle this problem using principal components regression. So the first thing to do is to fit the principal components model. So I read in the Boston housing data, which is on Sakai for the other lecture. I forgot to put it on for this lecture. I'll try to do that when I get back to my office. I fit a principal components model to the first 13 columns of the data. The 14th column is the response variable, the median home value, so I don't include the 14th column. I do only the first 13 columns. And then let's plot the scree plot and see what it looks like. Like I said, note that we did columns 1 through 13 rather than the entire matrix. As I said, that's because the 14th column is the response. We don't want to include that. Yeah. So, how many components to include? Look at the scree plot and make a somewhat arbitrary decision to include about 3. I mean, like I say, it's objective. You could make a case for 1. In this case, it looks like there's a little bit of an elbow once you go past 3, but as I say, it's subjective. So let's just go with 3 for the fun of it. Yeah, and just as an aside, rather than doing that little simplistic strategy where I eyeball the graph and say, it looks like we should use about 3, it's better to do cross-validation or something like that to choose the optimal number of components, but that's a headache to implement. I'm not going to ask you to do it in this class. If you're ever doing principal components regression, you're not sure how many components to use, then contact an expert who can help you with the cross-validation and so forth. And so now we fit a regression model to this data. First of all, we calculate the principal component scores for this principal component model using the predict command. Then I fit a linear regression model to predict the median value of each home based on the first 3 scores in this principal component model. See, when we do that, all 3 of the first 3 scores are extremely strongly associated with median home value, and just going off of R-squared is an imperfect of a metric as that is, we're explaining about 63% of the variance in median home values using those first 3 components. If our only goal is predictive accuracy, that's not too bad. And principal components regression, it's a possible alternative to lasso or ridge regression. If you have collinear data or high dimensional data, which approach is best to use, lasso versus ridge versus principal components regression? It really depends heavily on the problem, and a lot of it's motivated by what you're trying to do with the data. I mean, principal components regression has the same drawback as ridge regression in that you need every single one of the predictor variables to predict the response. I mean, granted, you're basing it off of like 3 principal component scores rather than 13 raw variables. You need the 13 raw variables to calculate those scores. Lasso is useful in that it can potentially reduce the number of variables that you need to predict the outcome. But if all you want is predictive accuracy, then the optimal method varies from problem to problem. Your best bet is just to try a couple different methods and see which works the best. And I'll note in passing that if you have high dimensional data with a large number of irrelevant predictors, principal components regression can work poorly. In fact, I wrote my doctoral dissertation showing that specifically microarray data, standard principal components regression works poorly, and you can do better using certain alternatives. Another common use for principal components analysis among bench scientists these days is if you ever do a genome-wide association study. Hopefully most of you are familiar with the basic idea behind genome-wide association studies, or GWAS studies as they're called, but the basic idea is that you genotype people at a large number of markers, usually somewhere around 1 million, and try to identify markers that are associated with the disease of interest. The intuition behind it, if hopefully most people in this class have taken basic biology, but the intuition behind it is that if you sequenced a person's entire genome, as of right now, that's still prohibitively expensive. People say that within 5 or 10 years that'll become standard and GWAS will be obsolete. But right now, we still can't afford to do that typically. So rather than looking at all 9 billion base pairs or whatever it is, we just select about 1 million of them dispersed relatively evenly throughout the genome, such that if there's a particular locus that's associated with the disease, that particular locus most likely will not be included in our study. There'll be a marker that's so close to that locus that we'll be able to find the association with that marker because it'll be in really tight linkage disequilibrium with the locus. If you didn't know what I'm talking about, don't worry about it. The basic idea is we're just trying to find genetic markers associated with the disease. And one issue that you can get into when you try to do genome-wide association studies is this issue called population stratification. That is that if you're familiar with basically the history of the human race, the consensus right now is that Homo sapiens originated in the Rift Valley of Africa and then slowly spread throughout the rest of the world. But like African Americans that stayed in Africa tended to interbreed with one another, whereas Europeans ended up in Europe, tended to interbreed, and you got Asians, Native Americans over here. Each of these groups became somewhat geographically isolated and started to interbreed with one another, and as a result, that had some subtle effects on each person's genes. This illustration here is, or this little graphic here illustrates this process called genetic drift to show how this can happen. Let's say that we have this population of flowers here, where some flowers are red and some are white, and the color of the flower is a simple dominant Mendelian trait. If you have at least one big A allele, then the flower is red. If you have two little A alleles, the flower is white. And let's say in this first generation of 10 flowers, only 5 of the 10 leave offspring, the flowers that are illustrated in white. These other red flowers with the green background, they don't leave any offspring. So in this first generation, the frequency of the big A allele is 0.7. 7 of the 10 plants, or I shouldn't say that, if you take the total number of big A's divided by the total number of alleles here, it's 0.7. Likewise, for little a, it's 0.3. Now in this generation, you'll notice there's more white flowers than there were in the previous generation. If you calculate the allele frequencies, now big A only has a frequency of 0.5, and little a also has a frequency of 0.5, results in more white flowers. In this case, let's say only 2 of the 10 plants leave offspring. These 2 pink flowers here, note they're both big A. End up over here, the little a allele is now gone. All future plants will have pink flowers because the variant that caused the white flowers just disappeared. Now, with humans, you typically won't see this drastic of change from one generation to the next. So number one, populations of humans tend to be much larger, and usually a higher percentage of humans leave offspring. So if allele frequencies change at all from generation to generation, the change is very small. But nevertheless, if you have 2 isolated populations that don't interbreed with one another very often, over time, the allele frequencies will start to vary from one population to the next, and over time, some of the alleles will disappear from some populations entirely. In fact, like Europeans, for instance, typically have less genetic variation than African Americans, just because Europeans was a relatively isolated population throughout most of its history. They intermarried with one another. Over time, some of these alleles disappeared, whereas African Americans, since the human race originated in Africa, have lost relatively fewer numbers of these rarer alleles. And this creates a major problem if you try to do genome-wide association studies. Now let's say you have a disease that's more prevalent among African Americans, like I think prostate cancer is a good example of that. That, as I noted previously, that just due to genetic drift, there's a large number of SNPs out there where the allele frequency is different between African Americans and Caucasians. So if you do a study that includes both African Americans and Caucasians, every single SNP whose frequency differs between African Americans and Caucasians will pop up as significant, which is bad because basically all of your results will be hopelessly confounded, that you can't tell if the SNP is associated with the disease. You can't tell if it's really associated to some genetic change that increases your risk of the disease, or if it's just a marker for the person's ancestry. It's actually race that's driving the difference between those with the disease and those who don't have the disease. I mean, I think I told the story earlier this semester of one time I messed this up when I was playing with the GWAS study myself, that I just got a data set that I did not realize included both Caucasians and African Americans, and I just mindlessly went away analyzing my GWAS, just immediately got like 5 billion hits, a number of which were extremely strong. I'm like, oh my gosh, I found all these genes that cause arthritis. I'm going to be like on the cover of Nature Medicine or something. Well, then somebody pointed out to me that it included African Americans, and I just found a large number of genes that tell you whether you're black or not. So yeah, that was kind of a downer. But, so, to avoid problems like this happening, what can be done about it? This is where principal components analysis comes in handy. Recall that the first principal component is chosen to explain as much variation in the data as possible. And so the hope is that since genetic drift causes variation in a bunch of different loci, that you can, like the first principal component will correspond to race in some sense. And if you adjust for that component, then you can eliminate the effects of ancestry/race and be able to identify SNPs that are actually associated with the disease of interest. So this is a plot I found online where they plotted the first two principal components of the data from a GWAS study, and this was just among basically Caucasians. So we're not even including African Americans here, or Asians, or anything like that are far more different genetically. But even among Caucasian populations, you can see the principal components captures variance between different national groups. We have Americans, then Russians and French a little bit further down, and see like some populations that tend to be more isolated a little bit further apart. You have the Ashkenazi Jewish population here, which tends to interbreed with itself more than the U.S. population as a whole. Then you have like Palestinians a little bit further out. Furthest out is Mozabite, which I don't know what that means exactly. But the point is, even among Caucasians, you can capture variation in ancestry by looking at the first couple principal components. So that suggests that rather than just identifying SNPs that are associated with the disease, what you can do is find SNPs that are associated with the disease once you take these first couple principal components into account. The intuition is that says that even once you take into account the fact that African Americans have different allele frequencies than Caucasians, this SNP is still more common among those with the disease than those without the disease. And for practical purposes, what that means is that you fit a regression model to predict whether or not they have the disease based on usually the number of minor alleles of the SNP plus the first couple principal components. And then in that regression model, you look at the t-statistic for the SNP, see if that's statistically significant. There's a software package called Eigenstrat that does this. I assume if any of you are actually doing GWAS in your labs, then you're very familiar with this. If you did need help with it for some reason, you're welcome to email me. Oh, things to remember for today is the principal components analysis can be used to reduce the dimensionality of a data set. If can take a large number of collinear variables and reduce them down to a smaller number of principal components that capture as much information as possible about the full data set. It's useful in a wide variety of situations, although it's difficult to sit there and enumerate situations where it would be useful. This is more just intended to tell you what principal components means if you ever encounter it in a paper or whatever, if you ever need to use it in your own work. And here's the new R commands today. PRComp will compute principal components, regressions, and there are also plot and predict commands if you want to plot a discrete plot or calculate the scores. Any questions about anything today? Okay.